Alright guys, this is the second video for the immune system. And so we're going to start off now talking about adaptive immunity. And so if you remember from the last video, we discussed adaptive immunity is specific. So it is going to be targeting specific diseases, bacteria, viruses, that sort of thing. And so each of these cells will be specifically tuned to target one particular disease, okay? And so with this section, we have a couple terms to introduce, all right? So our first term is an antigen. So an antigen is any kind of molecule that causes an immune response, okay? And so these antigens are going to be the target of the immune system. So the antigens are going to be what those cells of the adaptive immune system are going to be looking for. They're going to be looking for specific antigens that are associated with specific diseases. All right, and then we're also going to talk about antibodies. So antibodies are proteins that bind to antigens. Okay, and so they are made by B cells. All right, we're going to talk in detail about B cells and how antibodies work. And so remember, antibodies just circulate within the blood and the lymph, looking for some sort of antigen to bind to. All right, and so then our next term is probably new to most of you. All right, so that is a major histocompatibility complex, or an MHC. Okay, and so these are just membrane proteins, so they're going to be on the surface of a cell, and they typically have some sort of groove in them that's going to fit an antigen. All right, and so here, right in this picture, this would be our MHC, and then this would be the antigen. So it has some sort of antigen bound to it that it's holding. All right, and so there are two types of MHCs that you guys need to know. The first one is an MHC class 1, or we just call it an MHC1. These MHCs are on the surface of all of your body cells, and they can have two possible functions. So generally, if a cell is normal and healthy, they will function as a ID for that cell, saying it is belonging to the body, right? So it's that self-ID tag, okay? And so this would be in a healthy cell, okay? However, if there's something wrong with the cell, if it's infected with a virus or if it's cancerous, that MHC class 1 will actually present an antigen that says this cell is infected or this cell is cancerous. Okay, so they're on all of our body cells. Every single body cell would have one, and they'll either say this cell is normal, healthy, and belongs here, or this cell is infected and cancerous. Okay, and then we have MHC class 2. So MHC class 2 proteins are present on cells that are called antigen presenting cells. All right, and so we're going to talk about this in just a second, but they basically act like wanted posters. Okay, and so these antigen presenting cells will find some sort of pathogen, and they'll present the antigen from the surface of that pathogen to other white blood cells, saying, you know, this is the invader in the body, this is what we should be looking for, and then those cells can go out and fight that infection. All right, so let's talk about those antigen presenting cells. Okay, so these are going to be typically macrophages and B cells that act as antigen presenting cells. And so let's look at kind of the pathway of how they work. All right, so first they are going to engulf, right, or phagocytize some sort of bacteria or virus, some sort of pathogen. All right, so they're going to phagocytize it, surround it with their actual cell structure until they become completely surrounded and part of a vesicle within the cell. And then what the cell is going to do is it's going to merge that vesicle with the lysosome. All right, hopefully you remember the function of a lysosome is to digest things within the cell. And so then this plus the lysosome, all right, so now we have that cell, that bacteria, or the virus with all of those digestive enzymes of the lysosome, and that's going to break down that pathogen into a bunch of little pieces. And so then what that cell can do, this antigen presenting cell, is it can take a little piece of that pathogen, an antigen from it, and add it to an MHC2 protein. All right, so that's what we see here. So once it's made this MHC2 with this special antigen from this new pathogen that it's found, it will add that MHC2 to the surface. All right, and so now this antigen presenting cell has found something new, something that needs to be addressed by the immune system. It's made this MHC2, this wanted poster, it's put that on its surface, and so now it's going to travel to the lymph vessels, right? So it's going to enter a lymph vessel because it needs to go find an immune cell to show this to. So it's going to travel through that lymph vessel into a lymph node, all right? And once it's in that lymph node, it's going to find a T cell, and we'll talk about a little later specifically what T cell that is. But for now, we're just going to call it a T cell. All right, and so it's going to show that wanted poster to the T cell, and that is going to cause this T cell to become activated. Okay, so this is now an activated, that's why it's all excited, right? This is an activated T cell. So then that T cell can go and start the process of fighting off that foreign invader. Okay, so this is the process of how antigen presenting cells work. So this purple cell would be either a macrophage or a B cell, and it presents an antigen on an MHC class 2 protein. That's why it's called an antigen presenting cell. Okay, all right, and so then there are two more cell surface proteins that you guys should know. Those are T cell receptors and B cell receptors. So on the surface, like this T cell we were looking at up here, right? On the surface of this T cell, it has this little kind of orange, right? If I zoom in really quick so you can see it, right? It has this little kind of orange bracket here. That would be the T cell receptor. The T cell receptor allows that T cell to interact with that antigen on the antigen presenting cell. So it binds to the antigen. Okay, and specifically with B cells, and we'll talk about this when we talk about antibodies, the B cell receptor on the surface of a B cell is actually an antibody, but that antibody is what interacts with the antigen on that antigen presenting cell. Okay, all right, so now let's talk about our types of adaptive immunity. So there's two types of adaptive immunity. There's humoral, which functions through antibodies, right? And those are going to be made by B cells. So that system is going to involve B cells. And then we have cellular immunity, which is going to involve a form of T cells. So there's actually two different types of T cells. So let's go ahead and talk about what those two are. The first type is called a T helper cell. All right, and this T helper cell actually functions in most branches of the immune system. Okay, so it's also referred to sometimes, you'll see in your text, as a CD4 cell. That's simply because it has this extra protein, right, next to its T cell receptor. All right, so that's going to be a T cell receptor. Okay, so next to its T cell receptor, it has a CD4 protein, and that protein just functions to help, we'll add that up here, so it helps the T helper cell interact with the MHC2. Okay, So that's the function of the CD4, and so that is on T helper cells. T helper cells themselves function to activate other cells of the immune system. So they're going to interact with that antigen presenting cell, and then they're going to go and spread the word and activate other cells. Okay, so they're helping. They don't generally fight the pathogen themselves. They activate other cells that are going to do that. And then we have T cytotoxic cells. All right, these are called CD8 cells because they have this CD8 protein with the same function of the CD4. It helps that T cell receptor to interact with its specific antigen. Okay, and so these T cytotoxic cells are going to attack and destroy any abnormal cells in the body. All right, so now we've introduced our cells. We have our two branches, humoral and cellular. Let's talk in detail about the humoral response. Okay, so as we said, the humoral response involves antibodies, which are made by B cells. All right, and then we're also going to need our antigen presenting cells and our T helper cells to kind of alert that humoral response that there is a problem. All right, so let's talk about a couple general topics first. So with both the humoral and the cellular response, we're going to produce what is called memory cells. Okay, so T helper cells, T cytotoxic cells, and B cells can produce memory cells. And so they're produced after the first encounter with a new pathogen. All right, so like the first time you got chicken pox, these would be your primary response. Typically, the immune system is very slow for this response. So this is a slow response. It can take over 10 days. The antibody that's made, the specific class, is called an IgM. And this is where those memory cells are made. So the function of the memory cells is to speed up the response the next time you encounter that pathogen. So the next time you encounter chicken pox, you don't actually break out, with the itchy, chicken pox because those memory cells are there and they're going to eliminate that new threat immediately. Okay, so the secondary response, which would be the next time you encounter that pathogen, those memory cells are going to respond, and they're going to respond very quickly. All right, that's going to be within hours or days. So you don't go through the process generally of getting sick again, right? That's when you have immunity against some sort of specific disease, is when you have these memory cells that can respond quickly. All right, so this is the humoral response. It involves antibodies. So let's talk about antibody structure. So with antibodies, we'll typically be abbreviating them AB for antibody. All right, they have two ends on them. So on a typical antibody, they're usually drawn as kind of like a Y shape. All right, so this would be like a classic picture of an antibody. We're going to see they can have different shapes though. So with an antibody, we have the variable region, which is going to be on kind of the tops of the Y here. And that is going to be specific for an antigen. Okay, so this part, right, this variable region is going to be different for each specific type of antibody that is going to attack a different antigen. Okay, so antibody against flu would have a different variable region than an antibody against the chicken pox. Okay, and then we have the constant region, which is down here, right? It's the kind of vertical stick of the Y. This is what's going to determine the antibody's class and therefore how that antibody is specifically going to act to get rid of a pathogen. Okay, it determines kind of the function of the antibody. Okay, so let's talk about our classes. All right, so we categorize antibodies just with letters, right? And so if you want to remember all the different types of antibodies, it's going to be MAG, right? So we have antibodies that are M, A, D, G, and E. All right, so we're going to talk about each of those. And so we put IG in front of each letter. That just stands for immunoglobulin. That's just another name for an antibody. So immunoglobulin G, all right, will be the first one we'll talk about. So IgGs are the most common type of antibody, all right, and they have this kind of classic shape. We were talking about that Y shape for an antibody. They're abundant in the plasma. If you ever go and have blood drawn and they check your titer for a specific type of antibody, right, that they're checking for the presence of that IgG against something, okay? And these are the antibodies also that can cross the placenta, all right? So these are the antibodies that you know, if you get pregnant or, you know, your significant other gets pregnant, they're going to be passing on to the baby, okay? So those are IgGs. These are also the antibodies that will respond for that secondary response from those memory cells. And then we have IgMs, which are these big guys, all right? Remember we said IgMs are the first antibodies made during that primary response. And they're made because they can bind a ton of antigens, right? They can bind up to 10. So it's like we put together 5 of those separate Y antibodies, right? So each one can bind 2, so you can bind up to 10. That way you can, once the immune system gets going and you know, starts to attack during this primary response, you can eliminate antigens very, very quickly. All right. Then we have IgA. So IgA antibodies are found in secretory fluids in the body. That includes tears, mucus, intestinal fluids, and breast milk. So again, these could be passed on to a baby through breast milk. And they just help eliminate, you know, in the mouth or in the eye, that possibility of, you know, infection. And then we have IgEs. So, and IgE and IgDs are both going to be antibodies that are present on the outer surface of a cell. And so this is kind of what that would look like, right? So you have your cell, and then there's going to be a lot more than just 5 of these, but it would just be covered in antibodies all over the surface like this. Okay, so IgEs are found on mast cells and basophils. When they encounter an antigen, they're going to cause those cells to release histamine. Okay, that's our IgEs. And then IgDs are going to be on the surface of B cells. So remember we said those are the B cell receptors. Okay, good. And so you can receive immunity, humoral immunity, in a few different ways, okay? So a few different ways that you can get these antibodies. So you can get them actively. That means that your B cells are actually being activated by an antigen and they're making antibodies, okay? So they can be activated in an active natural way. This would just be like if you get the chicken pox, right? You encounter that antigen, your B cells make antibodies against it, and now you have that immunity. Or it can be active artificial. So this is how most vaccines work, right? You're injected with an antigen so that your body will make antibodies against it, and then you are immune to whatever that pathogen is. So usually it'll be like a broken down virus or bacteria, right, that just has the antigens associated. So it's not actually live. You're not generally going to get sick from it. And then you can get antibodies passively. So this is where you're going to receive antibodies. So if it's passive natural, that would just be the natural way of receiving antibodies from a mother, right, across the placenta or through breast milk. And then there's passive artificial. So this is where an individual is injected with an antibody. Right? So an example of this, that we talked about is RhoGAM. All right, so remember with RhoGAM, you inject antibodies when a type negative blood mother is having a positive baby, so she will not make antibodies against that positive blood from the baby. All right, so let's talk about how, this will be the last topic for this video, how antibodies actually fight pathogens. There's a few different ways that they can. So the first one is through agglutination, which we talked about with blood clotting. So that's just by causing that antigen, and frequently the bacteria or whatever virus that's associated with them, to clump together. All right, and so that's kind of what it looks like here, right? Only we'd have a lot more antigens and antibodies, and it just forms these giant clumps, and then phagocytes can come in and eat those and dispose of them. Okay. Also, antibodies can neutralize pathogens. Okay, so they can bind to and cover up harmful regions on the pathogen, or prevent them from, if it's a virus, from infecting a cell. So they kind of neutralize it so they're not harmful, and they kind of, almost, it's like, they're detaining it in handcuffs, so it's not going to harm anything until the phagocytes or macrophages can come in and get rid of them. Okay. And then, as we discussed with complement, they can opsonize, all right? So opsonization, they can coat the surface of a pathogen, making it easier for a macrophage to phagocytize it. And then lastly, they can activate that complement protein system that we talked about. Okay, so that's it for this video. So in the next video, we're going to go through the steps of the humoral response and the steps of the cellular response. And then I'm going to go through and kind of put it all together in one big picture for us, so we can go through visually and see how these systems work and how they work together. And then I've made a worksheet out of this for you that's just practice. It's not an assignment, but that you can do to kind of practice this process and the terms and stuff like that. Okay, so we'll do that in the next video.